Danielle and my mentor in my lab is Dr. Hua. I'm currently a senior at Choi High School. My lab main research focus is the role of NHEH in the intestines. My lab question is, how can we thoroughly identify wild type and knockout mice in order to find the NHEH protein? What my lab is mainly trying to find through its research is how NHEH, the protein, and mucin production are connected. With this, we are able to see how NHEH is directly involved in the mucin production, and this gives us hope in finding a cure to patients that suffer from ulcerative colitis and other diseases that are in the intestine. In order to find connections between NHEH and mucin production, we first have to find NHEH gene marks in the mice. And to do that, we do a technique called genotyping. It tells us the genetic makeup of the mice, and that allows us to see if the mice have NHEH protein. We can differentiate this gene by identifying if they are wild type or knockout. Wild type meaning that they do contain the NHEH protein, and knockout is that they don't contain the NHEH protein. What we mainly use for this technique is called a PCR technique that allows us to break down the DNA and copy it many times over and over. We then create a gel called agarose gel, which once it solidifies, we put it into a container with a TIA solution. Then we put it to a voltage. The solution will run to a positive charge. Then after that, we put it in a machine that takes and it will show us a result, whether it is knockout or wild type. Now our results came out great. As you can see, it came out clear, and with this information we can see that there are wild types and there are what we call heterogeneous, when one parent has the NHEH gene and the other does not. We mainly use heterogeneuses as breeders. After genotyping, we could further do experiments with wild type and knockout mice to be a step further to discovering the importance of NHEH in the mucin production. Thank you.